Several news sections present information related to the incidence of cancer in Indonesia, especially lung cancer, which is the highest case of death and has caused more than 26,000 cases of death. The data shown by the Ministry of Health in 2023 is in line with the data released by Globocan 2022, which shows that lung cancer in Indonesia is the second highest incident cancer, with the highest number of deaths compared to other types of cancer. Current cancer treatments such as surgery, chemotherapy, radiotherapy, immunotherapy, and targeted therapy have not been fully effective in inhibiting the growth of lung cancer cells, often causing cancer, recurrence, and toxic effects that adversely affect the health of patients, which can lead to increased case of death. Literature studies show that failure in lung cancer treatment is often caused by overexpression of certain genes that significantly affect lung cancer treatment. The EGFR biomarker was found to be overexpressed in 62% of non-small cell lung carcinoma cases, resulting in poor life expectancy for patients. The overexpression of EGFR and HER2 genes activates the PHK signaling pathway as well as the RISE pathway, which in turn activates downstream signaling, leading to increased proliferation and survival of cell cancer.
Therefore, innovation is still needed to find alternative therapeutic agent for lung cancer treatment. In response to this issue, we SMAN Negeri 1 Yogyakarta team, consisting of myself, Andri Dwiratmanja, and my colleague,
Muhammad Geovani, are proud to present our research title. Several aspects of natural materials is currently being developed as an alternative therapy, one of which is cancer. Several previous studies have suggested that herbal medicine have an important role in enhancing anti-cancer effects and reducing the toxic effects of conventional therapy. Some examples of herbs that are proven to help cancer treatment include Mars alba and Tuslago farfara. In addition, data shows that 77% of lung cancer patients often combine herbal therapy with medical treatment to strengthen the benefits of conventional therapy, one of the natural materials that has the potential to be developed in brown algae.
Based on this background, the formulation of our research is, at first, what is the compound content of profile of sargassum extract? Second, how to predict the gene target of metabolic compound of sargassum extract against lung cancer cells by bioinformatic analysis? Third, what is the interaction profile of metabolic compound of sargassum extract with the target receptor based on bioinformatic analysis through molecular docking approach? At last, what is the cytotoxic profile and effect on target genes based on bioinformatic analysis of sargassum extract against lung cancer? This research was conducted from July to January 2025. It began with sample preparation and species determination to verify the brown alga species to be used. The extraction process was carried out in Parametrical Biology Laboratory at UGM to obtain a concentrate extract. The obtained concentrate extract was then identified for its chemical compound using the GCMS method. The GCMS test results were further analyzed using bioinformatic analysis and molecular docking. To achieve our research objective, we conducted cytotoxic and PCR tests at the Parasitology Laboratory at UGM. After the research data was collected, data analysis was subsequently performed. From the series of research we have conducted, the results obtained are follows. In the determination process, it was confirmed that the sample used was in the species Sargassum lubricatum. During the extraction process, the weight of the concentrate extract obtained was 78.11 grams with a total yield of 22.32%. The basic principle of GCMS is analyzing the mass spectrum of vaporized component from liquid extract samples through ion fragmentation. The mass spectrum is evaluated based on retention time and peak height, followed by calculating the relative abundance percentage. The data show 86 peaks, representing 86 compounds in the brown alga extract. The compound with the highest abundance is hexadecanoic acid ethyl ester, found at peak 48. Bioinformatics convert biological data into computational codes, such as SMAN code, and analyze them using algorithm and database. Eight-gene slices were obtained with the K-gene consisting of EGFR, ERBB2, and PCI3K, with EGFR having highest interaction and highest score. These results indicate the potential of the kinase as key target in therapeutic research and development.
Next, the molecular docking method was performed to predict the binding between the ligand and the protein present in the lung cancer cells. Our MSTs were within a reliable range less than 2A, indicating a valid procedure. The docking results showed that arachidonic acid had a stronger interaction. Docking score -10.22, followed by neopentadine -9.17, and hexadecanoate acid ethyl ester -8.68. These compounds have potential as bioactive candidates for the future research. Next, we evaluate the anti-cancer activity of the extract by several methods. The first being the entity assay. The entity assay method was chosen to evaluate the anti-cancer activity based on cell family data, where the basic principle of the entity assay method is that living cells will naturally secrete the enzyme succinate dehydrogenase, which will then convert the yellow entity assay reagent to purple formation salts. The higher the activity of succinate dehydrogenase enzyme confined by the purple formation salt solution, which means high cells viability. Conversely, if this enzyme is inactive or the amount is low, the yellow color of the anti-TSI reagent will remain, indicating that the cells are dead. Based on the graph data, it can be seen that prawn alga extract showed a strong inoxidatoxic effect, significantly inhibiting the viability of A549 lung cancer cells by 30.6%. Next, we evaluated whether the extract could inhibit the overexpression of EGFR and HER2 genes. We chose the PCR method because the principle of PCR that can synthesize and amplify DNA segments exponentially in a short time, up to millions of times, is the reason we chose this method. From the graph of the results shown, it can be seen that treatment with prawn alga extract is able to significantly reduce the expression of EGFR and HER2 genes compared to the control group. There is a significant decrease in expression in both genes, which indicates that prawn alga extract has the potential to inhibit cancer cell growth pathways associated with overexpression of EGFR and HER2. From the research we have done, we get the following conclusion. 1. Sorghum SP extract contains 86 compound which can be seen from the peak chromatogram of KCMS with the compound serotonin acid ethyl ester is the compound with highest abundance. 2. Geoinformatic analysis showed 5 genes with the highest rank that are predicted to be the main interaction target in the anti-cancer mechanism of the main metabolic compounds of Sorghum SP extract are EGFR, RBP3, PI3KC8, TART, and TFF3. 3. Molecular docking of 11 compound with the highest abundance against EGFR protein with its native ligand complex econometinib showed a docking score of -8.38, which is lower than the docking score value with the main metabolic compound of Sorghum SP extract, serotonin ethyl ester, with a docking score of -8.68 and MSDLS value less than 2.8. 4. Sorghum SP extract showed a toxic effect against I514 lung cancer cell with a activity value after 48-hour treatment of 305.4 plus minus 11.7 micrometer and at doses a quarter and a half IC50 can inhibit EGFR2 expression.
The suggestion from the study is further research is needed to assess its cytotoxicity and selectivity against other cancer cells and normal cells. Second, in addition, it's important to explore the potential of prawn alga in combination with the chemotherapy drugs to understand possible additional therapeutic benefit and optimize cancer treatment strategies.